My name is Shireen Chan, and I am an associate professor at the Medical University of South Carolina, and I am also a co-founder of Neurowing Therapeutics. These are my notes on innovation. I teach at the university. Yes, I'm a professor. I have a research lab at MUSC, and I also do some teaching and service at MUSC. I didn't always know that I wanted to teach. I'm from Australia, and I did my undergraduate degree in science, and then I did an honours year, which is a research year, and that's how I became interested in mitochondria, which are the powerhouses of the cell. They make all the energy for the cell. And that's what I knew I wanted to do, whether that be teaching or research or anything like that. That's the path that I followed. Yeah. So I was born in a very small town in Western Australia. It's a small gold mining town, and I lived there for most of my life up until the age of about 26, I guess. Well, I also lived in the UK for a bit for my PhD. Then I moved to Research Triangle Park. Research Triangle Park, where I worked at the National Institutes of Environmental Health Sciences. That's a subset of the NIH. And I worked on mitochondrial research there, looking at mitochondrial diseases and looking at a lot of patient studies as well as animal studies and also in vitro studies in the test tube. And so then after that, I came here to work at MUSC. Today we are in the SCRA Innovation Building, the SCRA Innovation Building on Meeting Street in downtown Charleston, and this is where we have our labs. Lab 10 is where Neurowing Therapeutics resides. Neurowing Therapeutics is a spin-out company that we developed out of our research, our academic research from MUSC. We started it in 2015. And this was born out of a discovery that we made around about 2011 at MUSC. And so we discovered that small vitamin K-like compounds, basically they protect the mitochondria and they protect neuronal health. So they make the cells live longer. And they also seem to, basically where we saw that in animal studies, that they're efficacious for reducing seizures in epilepsy and also decreasing the level of neuronal loss in Parkinson's disease. And we've also seen that some of our compounds are really good for rare mitochondrial disease as well. And so that's how Neurowing Therapeutics was started, and that's what we've been doing since then. So our goal of our research is to develop these compounds for different neurological diseases. So we've got a platform of vitamin K analogues, and there's certain compounds that are better for epilepsy and certain compounds that are good for Parkinson's disease, for example. And so what we're doing now is we're starting off with epilepsy. We've got NIH funding, phase 2 SBIR business funding. And so we're now determining what the lead compound is to hopefully be used in humans. And our next disease is Parkinson's disease. And so we've got a compound that we want to tweak to make it better and also to hopefully develop it for human use as well.
How does that breakthrough happen? Did you know what you were going towards in that, or was it kind of
so here's the story. So basically, James and I, so we're co-founders, right? So basically we started in the Department of Drug Discovery at MUSC around the same time in 2009. And so he's a medicinal chemist and I'm a biologist. And we had talked about different research projects that we could potentially work on together. And so there was this little grant that was available at the time, a sector grant at MUSC. And basically, it required two people who were of different backgrounds to come together and collaborate. And so we devised a little grant, a little proposal that is to test HDAC inhibitors, different types of inhibitors on a zebrafish model of epilepsy. And so basically we thought that someone would have already done this by now, or at least one of the big companies would have done this by now, but no. So James's background is on these particular inhibitors. So we tested maybe 10 of these specific inhibitors, and one of them really worked. And so James, he's the chemist. So basically he saw that the central moiety of our lead compound, or a hit of that hit, looked like vitamin K. So we tested vitamin K, different versions of vitamin K, vitamin K1, K2, K3. And K3 works the best. So it's the smallest vitamin K. And basically, different vitamin K analogs were made. So they're very small. And we tested them on the zebrafish model of seizures. So basically, it's you've got these little baby fish in a plate, and you put a compound that you want to test. And then also you put a compound that will cause them to have seizures. And so what we can do is we can measure how much seizure activity they're going to have, how much distance that they move in the little well. And so what we saw was that these little vitamin K analogs, they seem to work on preventing them from having these seizures, which is really cool. And so the NIH has this free program to test compounds that you might have discovered in mouse models. And so these can be very expensive, these models. And so we interviewed with them, and they looked through their database and they didn't see any compounds that were like ours. So they tested them, and our best hit in the fish actually was the best hit in the mouse models for epilepsy that they had, which is really cool. And so because there's about 40% of the people who have epilepsy have medication resistance, which means that the current drugs on the market, they don't work for these patients, or they have so many side effects that you just can't take them. So it's a big population of people. It's about 1 million people in the US alone. So yeah, so that's how we got started. We have started to, by getting money from the NIH, as well as getting money from the SCRA, the South Carolina Research Authority, we've been able to take that hit and develop it and optimize it such that hopefully we'll find a compound that can be used in humans.
How do you go about getting the money, the funding?
How do you get the money? Yeah, you have to write grants, or at least that's what we've been doing. Yeah, most of our money is from grant funding, and it's been from the National Institutes of Health. They have an institute called the National Institute of Neurological Disorders and Stroke, and they're very interested in neurodegenerative diseases like epilepsy, like Parkinson's, Alzheimer's, yeah. And they have been very, very supportive of us. So we've gotten 2 grants from them, 2 business grants. And the second grant that we recently got is for 2 years, and it's for $1.5 million. So it's a substantial amount of money that's enabling us to get to that next step. We were very lucky in that MUSC, the Foundation for Research Development, has been very supportive of us. So one of the things that you should do is get help. So we had never written business grants before. We've written academic research grants, and it's a whole different ballgame. So you really have to understand what the product is and think more business, which we are not. Well, we were not trained as business people. And so we had to really focus on the product and the milestones and the sorts of things that they're more interested in, the people reviewing the grant. So we had a lot of people read our grant, and also people to help us write the grant too, or critique the grant. Yeah. So the other cool thing is that in our department, we've got lots of people who have spun out their research into different businesses. And so they have gone that route, and so they've been very nice to us, letting us see their grant proposals and giving us advice as to where we're being too sciencey, for example, because that's what we generally do. Yeah. So the grant process can be very long, not just writing the grant, but also once you submit the grant, it takes about 9 months to know whether you've actually got the grant, and you might not have got it at that point too. So it's a lot of being patient too, and hoping that you have the money. But yeah, we've also started talking to a lot of VCs and other people who also have different types of money. And that's something we have to learn because that's very new to us. We've gotten money from MUSC. We've also gotten money from the South Carolina Research Authority, the SCRA. So we've gotten for our first business grant that we got from the National Institutes of Health, they gave us a matching fund, not matching, but basically they gave us a bunch of money to help us with our research, And they've also given us other help. So they own this building, for example, and so this has been a really great space for us.
This building gives you access to tools that you would...
That's right, yeah. So the equipment and the tools and the people, it's been a nice space. This facility here, the SCRA Innovation Building, has been really quite good for us. Not only do we collaborate with some of the people that we share space with, but we also have been talking to other companies, talking to them about how they have garnered money and how they do their day-to-day business and that kind of thing. So it's been a really good way to learn. The one thing that I was thinking about to answer your question about a failure that we've overcome, I'm not sure if I've overcome it yet, but a few years ago at MUSC, there was a Pri-Med program that was run for people who are starting their own startup companies out of their academic research. And the first day, I think it was the first or second day, because it was a weekly course. And basically we had to go and give a 10-minute pitch. And everybody's was terrible, basically, because we always focus on the science. And we're always talking about the nitty-gritty and all the little things on our graph and whatnot. And so basically what we learned over the course of the next few weeks, as well as from our mentors that were part of that program, who are still, some of them are our mentors today, basically we've learned how to condense that down. I don't know if I'm perfect yet, I would say no, but I've gotten better at trying to answer what I think the audience wants to hear, and not so much the nitty-gritty, too verbose type thing. We know from the literature that there's a certain type of vitamin K that is absolutely needed for neuronal health. And the way it works is that it protects the mitochondria, protects those powerhouses that make the energy for the cell, right? And so that is not the same type of vitamin K that we get from our diet. So vitamin Ks you can get from green leafy vegetables, you can get it from animal products, you can get it from fermented foods. So you take it into your body, but then these vitamin Ks need to be processed and they need to be transported. And once they're into the specific neuron itself, it needs to be also metabolized as well. So what we see is that our vitamin K analogs, they're really small. So they directly penetrate the blood-brain barrier. They get into the cells in the brain. And so that's how they work. So instead of having to do all that transport and metabolism, it goes straight to the brain.
Do you feel like there might be more applications of your method?
Yes, so some of the genetic links to Alzheimer's seem to be associated with vitamin K. And as you get older, your pools of vitamin K actually get smaller, right? But one of the things that links all of these neurological diseases is that the mitochondria tend to fail in a lot of these diseases. And so if you can protect your mitochondria, protect them, enable them to make enough energy for health. So the brain requires a lot of energy for its function, right? And the mitochondria, unfortunately, when they become dysfunctional, they also produce a lot of oxidative stress, a lot of free radicals and those kinds of things. And so these compounds also help to decrease the production of those kind of compounds. So our goal right now is to find a partner, to find an investor, which would be great. So we've gotten, if we go the traditional NIH funding route, we've gotten the phase 1 business grant. We've now gotten our phase 2 business grant. The third one is a phase 2B business grant. And this is for 3 years, up to $3 million. But one of the provisos on that is that you need to find someone who will match those funds. And so, that is our next goal, is to find someone who can find an investor who will help us with this.
When you were a little girl, did you know you wanted to be in science?
That's a good question. So when I was growing up, I was surrounded by science. My dad was a chemist and my mum's a nurse. And whether I was going to end up in science, I don't know. But that's my background. I think it was easier for me to get into science just because I was surrounded by that, yeah. Basically, I moved from the other side of the world to Charleston, from Perth to Charleston, and that's a long ways. And but the reason for me moving is because I'm so interested in mitochondria and how it affects human health. So I was doing my undergrad degree, and then I did this research here with Dr. Jim Cummins, who in the vet school, and basically he calls himself a spermatologist. Let's just say that. So basically, what we were looking at was looking at the health of human sperm and understanding whether there were differences in mitochondrial health. And also because the mitochondria had their own piece of DNA, and we were looking at the state of that DNA, whether it was damaged in different groups. And he's my mentor to this day. I still go back every time I go to Australia, I go to see him and we chat. And but basically, I wanted to go to North Carolina because the enzyme that makes the DNA in human cells, or in vertebrate cells basically, the mitochondrial DNA. So what happened at that particular point in time is that the first human disease mutations were found. And the guy in North Carolina that I worked for, Dr. Bill Copeland at NIHS, his lab was the first to clone and characterize that gene. And so it was a great time to go there because I was able to learn a lot from his lab. So understanding about mitochondrial diseases, understanding how these mutations work to cause disease, looking at different animal models. And this is why I started to use zebrafish in Charleston, because I think that it is a good complementary model to using mouse models. So mouse models, they can be very expensive. They take a long time to develop disease. Sometimes they don't even develop the disease that you're looking for. So the fish, they produce hundreds of offspring, and you can look at the embryos in a plate, in a dish, and you can treat them just like cells in a dish. And so this is one way in which we not only understand about the disease and how it progresses, but also how we do drug discovery as well. So it's a new way of doing drug discovery. Instead of looking at a single cell in a dish, we've got these little baby animals in a dish that you can use like cells in a dish, yeah. But they have a working brain liver, for example. So in terms of the business, we have two mentors right now, or two people who I would consider our biggest mentors right now. And we were lucky to be introduced to these people, actually. So both of these people, they worked for a big pharma, and they decided to retire here in Charleston. So that's a lucky thing for us, that they decided to come down to Charleston, and that we're introduced to them, and they are so supportive of us. They've been imparting their knowledge onto us. They've helped us with grants. They've helped us with our pitch deck. They've helped us to be introduced to certain people that we would never have been introduced to otherwise.
Especially with what seems to be a years-long approach at trying to figure things out, at a microscopic level. How do you maintain that innovation?
So it's hard. So innovation takes money. It takes ideas, and so we have lots of ideas. And so currently we're trying to expand our platform, like I said. So we know that our compounds work in different disease subtypes. And so we're concentrating on our first disease first, the medication-resistant epilepsy, and then the and then Parkinson's disease, and then the mitochondrial diseases, the rare mitochondrial diseases. And so it's hard, yeah. It all takes money, yeah. Keep an open mind. I think that if you have an idea and you think it's a good idea, just go for it. If you can do it, go for it. And that's basically what we've been doing. We, as long as we had the funding, we basically went for it and tried new ideas. Yeah. So what motivates me to go forward is that if an idea sounds right, if it sounds exciting, and it sounds like a good way forward. Right now, what we're trying to do is I've been doing a lot of pitching. And so there are pitch contests, like we got accepted into SE Bio in Atlanta last year. And so I pitched to the group there. And that was great. In fact, we got some people who were very interested, no money yet, but basically at least we got our name out there. And then next month we've been accepted into the CED Life Science Conference in Research Triangle Park. And so hopefully we'll get something out of that too.
What's one very indispensable tool?
My computer and my cell phone are the generic answers. I think that everybody uses those these days. I travel with my computer, as you can see, and my cell phone, and I can't live without them. One funny answer that I was thinking about what I couldn't live without, and I think that it has helped me at least, is my accent. People always come and talk to me, or at least they are more interested in talking to me after I've opened my mouth. So that's been helpful to make conversation and get things started. I lived in Charleston for 10 years now, so I came here in 2009. And I like living downtown, and MUSC is downtown, and this place is downtown, so it's very easy to walk around everywhere. I really like that. I like not having to drive and park. I like the food scene downtown. I like to eat, and yeah, there's lots of events downtown. It's kind of nice. Yeah, I have got one good friend who's I would say is a Charlestonian, and he's introduced me to many native Charlestonians. And yeah, they like the good life. They really know how to party. They really know how where the good places to eat, and yeah, they're very social, and I like hanging out with them. I'm Shireen, and those were my notes on innovation.